Muito obrigado, Viviane. É verdade, eu entendi tudo, mas infelizmente eu não vou dar minha palestra em português. Desculpa, talvez próximo ano, talvez. But I'm going to switch to English, and I hope that you can follow my British accent. So I'm going to talk about a new project that we've been working on in the lab to try and address the question, can we identify a unique marker on latently infected cells? And if we can identify such a marker, can we then use that marker as a target to kill them? But before I show you our data, I'm going to give you some more background about latency. You'll have heard some of this yesterday, but I'd like to reinforce it. For those of you who haven't yet visited Washington, our department is Microbiology, Immunology, and Tropical Medicine at the George Washington University, which we abbreviate to GW. And we're an urban campus that's nestled in the heart of Washington, D.C. This is where we're located. We're just by a metro station and by some really great restaurants. We are also pretty close to the White House, and so we feel that we are close to power, or the seat of power. It doesn't always translate into anything useful, and it depends on the occupant of this. But we're also close to places like the Lincoln Memorial on this beautiful mall, so we can go and get inspiration and think about the good side of politicians. That doesn't take very long to think about. So my department, Microbiology, Immunology, and Tropical Medicine, is actually quite an interesting and old department. And Walter Reed, who was instrumental in working on yellow fever, was professor of bacteriology and microscopy in 1893. He's a really interesting person. He finished his medical training at the age of 17 and was the youngest medical student, I think, until Doogie Howser made his appearance on TV. Our department is in this building, Ross Hall. We have open labs. We have these nice collaboration spaces where we can talk about science or we can talk about football. And we have this area that's called a kitchen, but actually turns out to be a bar. In terms of research, we have two large areas that our faculty are interested in. One is the Research Center for Neglected Diseases of Poverty, and we're interested in understanding pathogenesis of parasitic diseases, including helminthic worms, and toxoplasmosis as well. And then I lead a center which we ambitiously call the Research Center for the Cure and Eradication of HIV, and we are interested in promoting studies on basic science related to cure research. So you'll have heard yesterday that there is persistence of HIV on antiretroviral therapy. We know that antiretroviral therapy in people who can tolerate it are able to suppress HIV to levels that are below the level of a detection in an assay. But ART cannot completely eradicate HIV from a person's body because of this persistence in viral reservoirs. And these reservoirs consist of HIV-infected cells in which HIV DNA has been integrated into the host cell genome, but are not actively producing virus. And this is the phenomenon, as you will know, called HIV latency. And those latent infected cells can be activated, and that cell can then produce an active infection. So after antiretroviral therapy is started, those cells can survive and are dormant for a long period of time while the patient is on suppressive antiretroviral therapy. But we know when antiretroviral therapy is interrupted, in almost all cases, virus will rebound. And that is considered to be an indication for the presence of these viral reservoirs. And so this is what necessitates the need for infected people to be on lifelong antiretroviral treatment. In thinking about what we may want to do and use this word cure, there are two possible scenarios in general terms. One could be this sterilizing cure, where as the viral genome is irreversibly incorporated into the genome of every infected cell, every single HIV-infected cell has to be removed from the body. There would be a complete removal of HIV-infected cells. In contrast, a functional cure would be one where HIV is not completely removed from the body, but is controlled in a way that makes it remains quiescent even when you take away antiretroviral therapy. We don't yet possess a way of making the virus remain quiescent in the absence of ART, or to completely eradicate every cell. But we think that there is a case of functional cure in Timothy Brown, also known as the Berlin patient. And his is an interesting but complicated case that I think we've talked about here in the past. But this, obviously, from a virological and immunological perspective, is highly complicated. So within an individual who is infected, we have to think about potential body compartments where HIV reservoirs could exist, different cell types, status of the virus, and status of the infected cell. So in terms of cell types, we know that there are a lot of different cell types that HIV can potentially infect. And while there is some suggestion that reservoir can be found at different amounts in some of these cells over others, I think it's still not completely identified the proportions or the full spectrum of where the reservoir is existing. And then we normally are accessing peripheral blood cells, but the whole of the rest of the tissue is probably much more important in relation to identifying areas where HIV reservoirs could persist. In terms of the virus, the virologists have a lot of interesting discussions about replication competent and defective different types of viruses. And then the activation status of the cell is also extremely important. So this provides a very complicated background in which to think about the reservoir. We know that the reservoir is established very early on in HIV infection, and the non-human primate model studies are helping lead the way in identifying more of this. And in humans, this was illustrated, I think, by the case of the Mississippi baby that excited a lot of HIV research when it was thought that this infant was functionally cured. She had contracted HIV at birth from her HIV-positive mum, and she had been placed on a highly active retroviral regime 30 hours after birth, so very quickly. The treatment was stopped after 18 months, and HIV didn't come back for a very long period of time, and ART was not restarted. But over 2 years later, HIV then became detected. So this was an example of an early aggressive antiretroviral therapy that, when stopped, led to a prolonged period where virus didn't return. And obviously there's a lot of interest in trying to understand why that virus actually came back. But it did indicate that this reservoir is established at an extremely early stage. We know that the reservoir is very stable. So long-term follow-up studies of patients show that the latent reservoir decays so slowly that over the course of a normal human lifespan, eradication with antiretroviral therapy would be practically impossible. And now, from an older study from Bob Silicano's group, we could see that there's this decay which would take 60 years, but newer information has suggested that it would actually take much longer than this. So it becomes a question of how is that reservoir sustained, because not all the cells might last that long. They might die off. So in an immunologist's view of a virological conundrum, I think that there are two main theories that either suggest that the reservoir is very long-lived or the reservoir is constantly refilled by ongoing low-level replication. And there is data to support both of these hypotheses. So I think it is not clear which way the reservoir is maintained or whether the reservoir is repopulated, and that's still something that is controversial. Again, I think the non-human primate models are very important in being able to access tissues that are harder to do in humans. So in addition to this sort of complexity and lack of understanding of the viral reservoir, we also know that HIV genomes themselves are extremely complicated and complex. We have ways of assaying different avenues of viral replication at various stages and looking at genomes. We have looking at GAG DNA sort of as an assay to do total HIV genome. We have integrated HIV genomes by allo-LTRs. We've got unspliced and multiply spliced RNA to look at viral transcripts. We have HIV proteins that are produced, viral particles, and defective viral particles, and then replication complicated virions. So the whole field is sort of populated by this complex and burdensome nomenclature that makes it a little hard to understand. And of course, we're interested in intact genomes and what actually goes on to make real particles that could then go on to infect and spread the infection. And in thinking about the size of the reservoir, I think it's now becoming clearer that if you can treat early, that the size of the reservoir is smaller. But it's hard to measure, and a number of different investigators have been trying to develop better assays to look at reservoir size, and we have these PCR-based assays, the qPCR, tPCR, digital droplet PCR, and then we've got these nice acronyms like TILDA, the TAT rev-induced limiting dilution assay, or QVOA, the quantitative viral outgrowth assay. And at the moment, many virologists believe that this QVOA is the best standard for measuring the inducible viral reservoir, but a number of studies have come forward to suggest that it's probably underestimating the true reservoir size. You take patient-derived resting CD4 cells, you do limiting dilution in number of replicates, you reactivate, so you get an activated CD4 positive cell, you add uninfected PHA blasts, you get amplification of the virus, you culture, and then you end up doing an ELISA. It's quite a tedious assay if any of you have done it. And then we think that this assay is probably underestimating the real reservoir by even up to 60-fold. And part of that is because not all intact replication-competent proviruses are induced when you reactivate the latent cells. So we have these different pots of different replication competence, and it makes this assay, while the best one we have, still inadequate for really measuring the reservoir. We think that approximately 12% of integrated proviruses are functional, but only a small fraction of those can be induced to produce virus ex vivo. From people, it gets even harder. How do you measure the reservoir in someone on suppressive antiretroviral therapy who's infected? You can try by measuring residual viremia in plasma. You can do tissue or cell-associated HIV, either by total integrated DNA or 2 LTR circles, or unspliced or multiply spliced or poly-A RNA. Isolated reservoir cells are activated in vitro, and we can also use RNA transcripts or inducible infectious virus to try and measure. It looks simple in a review, but it's complicated when you're doing it in the lab. In thinking about HIV transcripts in subsets of infected cells, In productively infected cells, there's integrated HIV DNA provirus, high levels of multiply spliced and unspliced RNA transcripts and virus production. And we think in these sort of true latently infected cells, there is integrated HIV provirus. There's no RNase transcripts or probably importantly, minimal unspliced RNA transcripts and no actual viral production. But there's this middle area, partially activated cells without virus production, where there's low levels of multiply spliced without virus production. So it becomes a complicated discussion and scientific challenge to really determine what cell is really latent and what is this partially activated cell. So I've been cautious in an English way and say technical challenges are abundant. In other words, it's really very difficult to work this out. Of course, as I mentioned, we generally access blood to do these studies, but that's probably only representing a small fraction of what cells might be, or where to find the cells that are in the latent reservoir. And tissue reservoirs are difficult to access in humans. And then there's this high burden of defective proviral DNA. And as I mentioned, even the best assay is only measuring a fraction of replication-competent viruses. I think important questions that come out of this is that we know that the latent reservoir doesn't only persist in resting memory CD4 cells, as originally thought, but in many other cell types. And so questions that come up are, what are the mechanisms of persistence? What's the difference between proliferating and non-proliferating latently infected cells? Are there unique features of tissue that promote HIV persistence or control? And then not all intact replication-competent viruses are inducible upon reactivation. So are they important at all? Do these defective forms matter for persistence and how? And are some viruses easier to induce? And what would that mean for viral latency? So you'll have heard yesterday about some of the therapeutic opportunities to try and change this setting by activating the latent reservoir and then trying to kill virus that comes out. And this has sort of been colloquially known as shock and kill. And shock and kill agents, approaches use these latency reversing agents, or LRAs. And common ones are HDAC inhibitors. You also have PKC agonists, P10 inhibitors, TLR agonists. But I think these are now agents which one of these alone probably is not going to work, and you have to potentially have them in different combinations. And those are studies that are currently being tried. There's an alternative hypothesis that Susanna Valenti has recently introduced, which is sort of a contrary to shock and kill. It's sort of a pacifist way of blocking and locking, where she postulates that after HIV infection, this is increase of viral load. You have these viral blips, you get latency. With TAT inhibitor in combination with ARVs, she is postulating a state of deep latency that might even occur even after treatment cessation. So shock and kill, block and lock, who knows what will be the next phrase. In vitro, we have been able to use a number of different latency models to look at some of these questions. And they can use either primary cells, transformed cell lines, or patient-derived cells. These have been used in a number of different ways. And so investigators have derived a number of different models of latency, and we have chosen to work with what's called the Sharon-Lewin model, named after Sharon Lewin, who's a wonderful investigator in Australia, who has developed a system that we use routinely. So the Lewin model uses primary resting CD4 positive T cells and relies on incubating with cytokines. And the Lewin model seems to have a wide range of abilities to respond to different stimuli. You incubate your cells after activation with CCL19 and 21, and then this model has been used extensively to look at some of these strategies. So with HIV replication, one of the most exciting things over the past 20 years has been the identification of host antiviral genes that normally are present, which can help restrict viral replication, and these are known as restriction factors. And one of the interesting things about the HIV life cycle is that there seems to be a natural way of inhibiting viral replication at various points of the life cycle. So SAM-HD1, APOBEC tetherin, SLAFN11. Over the past 20 years, a number of these important restriction factors have shown that the body does have ways of naturally restricting HIV infection. And Andre Raposo in my lab has been interested in host restriction factors and made some interesting observations, published now a couple of years back, showing, for example, that people who have HLA-B57 as in their haplotype have increased expression of host restriction factors, and that elite controllers have a selective expression of host restriction factors, particularly an increase in SLAFN11. And with Satish Pillai in San Francisco, who showed that in vivo these may have relevance and can be upregulated by interferon alpha. And in fact, most host restriction factors are triggered by interferon alpha. And so that also makes us remember about innate sensors. And when viral DNA is detected, a series of events is initiated that leads to the culmination in cell death. And the degradation of these DNA can occur in a number of different ways. Here we have TREX1, an exonuclease, which will degrade cytoplasmic DNA. You can also have CGAS-dependent sensing, which triggers interferon responses, and IFI16, which senses incomplete reverse transcriptates, activates the inflammasome, and caspase-dependent cell death, and you get interferon responses again, leading to cell death. So these sort of innate sensors are also important in protecting against HIV. And some host restriction factors also act as innate sensors. So tetherin, for example, could potentially act as a virus sensor in certain circumstances. An internalized tethered virus can trigger type 1 interferon expression via toll-like receptors. And then TRIM5-alpha as well can target the capsid for destruction and promotes the expression of certain genes by activating immune signaling pathways such as AP1 and NF-kappa B. So we came up with a hypothesis that the residual viral replication in latent cells during antiretroviral therapy could potentially trigger innate sensor genes, even if you have extremely limited viral replication in the upregulation of interferon-inducible genes. And some of these we know are restriction factors. So we postulated that if we could look in the model, in the Lewin model of latency, innate sensors and restriction models, we might be able to identify what might turn out to be a unique marker of a latently infected cell. And if we could identify something that was a unique marker of an infectedly latent cell, then we could have a potential target for the immune clearance of those latently infected cells. So we developed an in vitro Schauer-Lewin model where we took primary PBMCs and purified them. We treated them with CCL19 and then infected with virus. We sorted these into the various different populations. We got a latent population and then we reactivated it. And we looked by flow cytometry at GAG expression in the latent and the reactivated cells. We found, as we would expect, a very limited amount of GAG expression in the resting latent cell, and after reactivation, that went up. And then when we reactivated cells for a period of 3 days, we had a tenfold increase of HIV GAG and transcripts, including unspliced, multiply spliced mRNA as measured by qPCR. So there was a tenfold reactivation at the mRNA level. So we comprehensively analyzed the expression of a number of selected antiviral genes and most of the restriction factors. And we found differences in expression quantitatively of resting cells compared to cells that were reactivated. And in particular, with two, SAM-HD1 and IFITM1, we found that with IFITM1, there was a fourfold increase of expression in resting latent cells when compared to reactivated cells. Now, IFITM1 was interesting to us because it's a membrane-associated protein, and it's previously been reported with potent anti-HIV activities. So we decided to concentrate on IFITM1 as a potential marker for latent cells and to ask the question, could we target IFITM1 with an antibody to mediate ADCC to eliminate those cells? So I didn't know much about IFITM1 before we came across this, and it's interferon-induced transmembrane protein. Humans have different varieties, 1, 2, 3, 5, and 10. It's located on chromosome 11. IFITM 1, 2, and 3 have been shown to promote anti-proliferation, cell adhesion, and apoptosis. It's a 14 kilodalton membrane-associated protein with its C-terminus in the extracellular space and conserved intracellular region. It contains an interferon-sensitive response element and is very strongly induced by interferon alpha. Precise mechanisms by which it restricts HIV infections are unclear. It potentially interferes with endocytosis and also with GAG expression. Now, interestingly, to date, no viral antagonists from HIV has been described which works against this, and no one has hitherto, we believe, looked at the role of IFITM1 during HIV latency. So we wanted to do an antibody-dependent cellular cytolysis, the ADCC experiment, to ask the question, can we target IFITM1 on latently infected cells? And we started this by looking at latently infected cell lines. So we labeled latently infected ACH2 cells with an IFITM1 antibody or control and cultured them with PBMCs from healthy donors at different effector to target ratios. And we used a flicker assay to detect dead cells by apoptosis and monitored by flow cytometry. Here we found that at different effector to target ratios with the anti-IFITM1 antibody as compared to control, that the majority of cells were in the dead quadrant, suggesting efficient killing in vitro of this latently infected cell line through ADCC. And in parallel, we investigated whether cellular immune complexes that had been marked with IFITM1 antibody mixed with the latently infected cells led to NK cell activation. And we did indeed see activation both through CD107 and interferon gamma. So we looked at 7 different individuals. Sorry, no, I'm skipping ahead. We did this several times, and we looked at significant killing of ACH2 labeled cells with this anti-IFITM1 antibody compared to controls, and also saw increase in interferon gamma and 107. So the next question is to say, well, that was in a cell line, ACH2. What happens when you look at infected CD4 positive cells from ART-infected patients? To assess this, we sorted CD4 positive cells from a number of different ART-suppressed patients, and then we labeled them with the anti-IFITM1 antibody and then cultured them with effector cells in an ADCC assay. And the IFITM1 positive T cells were found almost exclusively in the fraction that were positive for the flicker marker, suggesting again highly efficient killing. And then to determine whether effector cells were able to work from autologous, we looked at autologous PBMCs, and they did not work as well as PBMCs from heterologous donors. So we then wanted to know a bit more about IFITM1 positive cells, but because there aren't very many IFITM1 positive cells, we would need a huge amount of cells to actually sort those. So we chose a different approach to look at whether there were any differences in the reservoir in IFITM1 positive cells by doing a depletion study and asked the question whether depletion of IFITM1 positive CD4 positive T cells would lead to a reduction in the HIV DNA pool. So we depleted IFITM1 cells from bulk CD4 positive cells from ART-suppressed patients, and then we quantified the total HIV DNA using droplet digital PCR. And overall, we detected a significantly lower level of HIV DNA in IFITM1 depleted CD4 positive cells compared to bulk CD4 positive cells. And we think that this might indicate that IFITM1 positive CD4 cells from ART-suppressed patients are those that harbor persistent HIV-1 DNA. We do not know yet whether this is a marker that is present on all latently infected cells. We don't know whether all IFITM1s have persistent HIV DNA. So there's a lot more studies that we are planning on doing, but we're excited about this preliminary data because what we think we've shown is that IFITM1 is overexpressed in latently infected cells. And it, at least in vitro, can be potentially a target for antibody-mediated cytolysis. And because we found that depletion of IFITM1 positive CD4 positive T cells would significantly decrease the pool of natural reservoir cells harboring persistent HIV DNA and inducible provirus, that we think it is worth exploring this further to understand whether this could be an alternative way of potentially identifying and targeting latent cells for destruction. So this was obviously work done by a group of people, really spearheaded by Andre Raposo, along with Claudia Brockmeyer, Miguel Mulder, Dominic Paquin-Pro, Phil Braley, Vinicius Cabado, recognize a Brazilian name there, Paul Znak, Alison Thomas, John Huang, and Brad Jones. I'd like to thank you very much for your attention.